let's move on to DNA sequence variation, which is a sort of natural continuation of what we've been talking about. Here's my disclosures again. So, yeah. Yeah. So the very good question. The question was, is hydroxyurea work in thalassemia? Not really, because it's never really been used. The prediction there is they're keeping up by making so many more red blood cells that the trade-off of shutting off red cell production would do far more harm than maybe a boost in the fetal hemoglobin. So you don't use it there, really only in, but we can talk more about it later. It's a good question. Okay, so we're going to talk more, what I want to talk now about is DNA sequence variation, the variation between all of us, which is extremely common and extremely important and the basis of all genetic disease, as well as the basis of all variation. If there was no DNA sequence variation, we would all be identical twins. And it's pretty obvious when you look around. I don't think we have any identical twins here. Certainly we wouldn't have more than one pair. So the very common forms of DNA sequence variation we call polymorphisms. And you're going to, hopefully, when I'm done here, really understand what I mean by the word polymorphism. And you're going to recognize the different types of variation that are common in DNA sequence. And we're going to talk about that. We've talked about it somewhat already with the globins as an example, but we're going to talk about the different types of classes of mutations that can disrupt gene function. And then I'm going to talk a little bit. I want you to think about, because this is becoming a huge problem now, when we get someone's sequence, and increasingly, when you guys are out practicing, it's going to be a whole genome sequence. We're going to find a lot of variation. If we take my sequence and one of yours, on average, there'll be about 3 million DNA sequence differences between us. Well, how do we know which ones to pay attention to and how not? How do we know when this is just one of those minor variations among people that doesn't matter, or that it might be causing disease? It's a fundamental problem that we don't always have a good solution to. Okay, so the fundamental paradigm, you're all comfortable and familiar with this. DNA, chromosomes, contain the gene, which gets made into RNA, which gets made into protein. And we've already talked about all this. One of the points, again, and I've sort of already mentioned this, about how unbelievably rapidly the field of genetics has exploded over the years. And as I told you earlier, this is where I went to medical school. And we really didn't know anything back then. Many of our concepts just turned out to be wrong. We had no understanding of any of the things I'm going to talk about today. Couldn't even imagine most of them, frankly. But when this slide was initially made in the textbook, this is sort of where we stood. And since over the past 10 years or so, lots and lots more has happened. We have a complete human genome sequence. We have a very lengthy index of all the common variations among humans. And we now have complete genomes of many different species. And we're going to talk about all that a little bit now and more in the next lecture. So you're all familiar with PCR, I assume. A way that we amplify DNA and routinely use. This is sort of a core method to take a small bit of DNA, even from a single cell, and get enough of it that we can then use to sequence, to look for mutations, to analyze in various ways. There are new methods to analyze that DNA, and I used to spend whole lectures talking about all the different methods we use to analyze DNA, to look at its sequence, to figure out other things about it. And I've totally eliminated all that from the lectures because it's mostly irrelevant now. Most of those methods you aren't going to use. The hallmark for most things now is sequencing. And the methodologies for sequencing are changing every day. So I'm not going to talk a lot about the methods. I'm happy to discuss if anybody wants to know how these things are done. But we're going to focus more on just what the result is and how you use it. And the methods they'll be using when you get out into practice is likely to be different. But one of the take-homes here is that we can look at, now we have methods for looking at large numbers of genes, the whole genome all at once. On a little chip like this, we can analyze the million or so very common variations in the population and see what any individual has at any of those positions. That's the basis for a lot of how we've approached complex diseases that we're going to talk about on Friday. We can look at how much RNA is made from any of the 20,000 or so genes. We can now pick up, and I'll show you some examples, large deletions or insertions or duplications of big chunks of the genome. This turns out to be much more common than we had ever imagined. In the book, they go over how standard Sanger sequencing works. This is rapidly becoming ancient history. This hadn't been discovered yet. This was the source of the Nobel Prize in 1978. Came out right around, it was discovered in 78. The Nobel Prize was later. Discovered right around the time I was in medical school. And this is now becoming old hat. We still use this for some simple diagnostic tests, but now we're using newer methods that are sequence billions of base pairs at a time. The very first research paper I wrote in the laboratory, I worked for a year and a half to determine 4 nucleotides at the beginning of the ribosomal RNA precursor in E. coli. And I know you've all always wondered this. It's ACUG. It's the beginning of the ribosomal. A year and a half. It was my first paper. Now, in my lab, routinely, we'll sequence 3 or 4 billion nucleotides in an afternoon. Totally astounding. If you think of the scale, we're not talking about a log or 2 log. We're talking about millions of times a greater a sequence per human. Totally astounding what's happening. It's totally revolutionized genetics and the way we look at genetics. Okay, so let's talk about DNA sequence variation in people. As I've mentioned, if we sequenced all our genomes, which we're going to do fairly soon, maybe there even is somebody who's had their genome sequenced, we're not the same. We're all different. I think you all probably knew that. We may be more different than you thought. Between any 2 individuals, we differ by about 0.1%, 1 out of 1,000 base pairs. And since the overall genome is about 3 billion base pairs, that's where I got that number of roughly 3 million differences between any 2 of us. I was talking to somebody about this at the break. Humans, our concepts of race and ethnicity, genetics is making all that look pretty silly. We're all pretty much the same, and we're all African. We all have left Africa, 50,000 some odd years or whatever it is ago, and our DNAs are fairly similar. The differences between if you take 2 people from 2 people from Africa and compare their genomes, and then someone from Africa and someone from Europe and compare their genomes, they'll all be about 0.1% different. You're no more like each other unless you're related to each other, as long as you're part of the diverse human population. If we take a chimp and a human, they differ by about 10-fold more. So we're remarkably like chimps, but we separate from them, a lot longer ago. They're about 10-fold more difference than we are, but this is the difference between 2 humans. Now, some of these variations occur within the coding parts of genes, change amino acids and all. More of them occur in between genes, in introns, regions that we used to call junk DNA, though they can't be junk. As we'll talk about, only about 1.5% of your genome actually codes for proteins. The rest of it, we don't really understand very much about what it's doing. Most of the variation is in the other 98.5%, but some of it is within the coding parts of the genes. Polymorphism just means that different forms of the gene can exist within the population. And geneticists have a very specific definition of a polymorphism, which I'm going to give you. I have to tell you, though, that the term is used incorrectly by almost everybody, including geneticists. And maybe I'm just too fussy about the definition, but I think it's important. It is an important concept underlying it. So a polymorphism just means that if we looked at humans, we find 2 different types of alleles. A mutation is any change in the DNA sequence from the original DNA sequence. Comparing ourselves to yeast or bacteria, that we may have evolved from hundreds of millions of years ago, almost everything in our genome is a mutation. So a mutation is sort of all in the eyes of the beholder. It does not mean that it causes disease, though very commonly in common usage, it's misused that way. You will hear very commonly many physicians or geneticists even say, is this DNA change a polymorphism or a mutation? What they mean is this a variant of no significance, or is it causing disease? And as the use of that is polymorphisms are mutations. They arose at some point as a mutation. So we shouldn't use it that way. But they can be a single base pair substitution. They can be deletions or insertions of a couple of nucleotides. They can be big DNA deletions or insertions or even rearrangements. So the strict definition of a polymorphism is given here. Now, there are DNA sequence polymorphisms. There are lots of other polymorphisms. Hair color is a common human polymorphism, whether you have blonde hair or dark hair, whether you have curly hair or straight hair. These are any variation in phenotype is a polymorphism. At the genotype level, a DNA sequence polymorphism, the formal technical definition is any variation in a DNA sequence where the rarer allele is more than 1% of the population. So if we sequence my genome and one of yours, and we find a variant in you that only 1 in 1,000 people have, that's a sequence variation that probably doesn't cause any problem at all, but it is not technically a polymorphism because the allele frequency is less than 1%. Okay. The other way to look at it is that the most common allele is less than 99% of all the alleles. Now, you can have variant alleles that are less than that are less than 1%, and it doesn't tell you whether it causes disease or not. This should technically be called a rare variant. And the usage, this is the reason I point this out is I think it's important in thinking about it. You'll see this misused all the time. We'll talk later about the term SNP, for single nucleotide polymorphism. People use that for any SNP, even one that's rarer than this that isn't technically a polymorphism. The more correct term is a SNV, a single nucleotide variant. Okay. So again, mutation is any change in the DNA sequence. It could be silent. It could cause an amino acid substitution. It might be neutral. It might cause disease. The fact that it's a mutation, that it was a change from the primordial sequence, does not tell you what that is. And again, as I've mentioned, this is going on all the time. Every generation, each of us has about 70 new mutations that were not present in our parents. Each cell in your body has a couple of mutations that was not in the cell that it came from. That's where cancer comes from. Cancer is a genetic disease. By chance, stochastically, where these mutations happen to hit, every once in a while they hit in a really bad place that results in a cancer. We've already talked about this concept of balanced polymorphisms, like sickle cell, and I'll come back to that in a minute. And I already mentioned this common incorrect usage of it is a mutation or a polymorphism. They're not mutually exclusive. And that's illustrated on this slide. So if we think of mutations, those that are normal, that have no real biologic effect that we know of, versus those that cause disease, and we think of whether we're going to technically classify it as a polymorphism, common or rare, all four combinations here exist. A rare normal variant is a rare variant. This is sometimes called a private polymorphism. It's a little bit of an oxymoron, but that's a genetic term. Private polymorphism, a rare variant, and this is a classic polymorphism. A rare disease-causing mutation, most disease mutations fall in this category. But there are some, like sickle cell anemia. If you go to parts of Africa where 20-30% of the population has a sickle cell allele, sickle cell anemia, the sickle cell allele, beta S, is a polymorphism. It's a disease-causing polymorphism. Factor V Leiden, the example I give here, is the most common risk factor for blood clots. About 5% of Europeans carry it. 1 in 20 of you sitting in the audience here probably on average carries this. That's a polymorphism, but it does cause disease. Okay. So let's talk about the different types of DNA sequence variations that one can see. The first we used to study years ago, and this has again almost become just a historic artifact, we won't talk about a lot, is an RFLP. Some of you may have heard of this. That stands for restriction fragment length polymorphism. Back in the old days, 80s, about the only way you could detect differences in DNA were using restriction enzymes and looking at the DNA on a southern blot. And if the DNA change just happened to change a restriction site, you'd get a different pattern. So early on, these were the only polymorphisms we could study. They're really single nucleotide changes most of the time. There are common variations in the genome that I'll illustrate in a second that affect repeated sequences, how many copies you have, either big ones, VNTRs, or littler ones, single nucleotide polymorphisms, or more appropriately, SNVs, single nucleotide variants we're going to talk about. And there are various insertions and deletions that can occur, and then these big variations. So this is a simple tandem repeat. These are a very common form of variant that we still use somewhat diagnostically. Here you have a stretch of sequence that's repeated. In this case, 10 copies. These are often unstable. Maybe once every 20, 30 generations, there'll be an unequal crossing over or polymerase slippage, and it'll get longer. So it turns out this position, you could have 10 or 11 or 12 or 13 or 14 or 15 different copies. Many different possible alleles here. That can be quite common, so it can be a polymorphism, and you get a different pattern in everybody. There are millions of these in the human genome, and they're extremely useful diagnostically. You could imagine, if you took one of these guys, any two people are not all that likely to have the same two bands. And if you used a panel of 5 or 6 or 7 of these, you could come up with a panel that you would have less than a 1 in a billion chance of two people matching. And this is, in fact, the core of much forensics now, is done with these kinds of markers. You can identify people. Identical twins will have the identical pattern, but other than identical twins, you can use enough of these so that everybody's different. Yeah?
Is this the same idea as microsatellites?
Yes, these are microsatellites, another name for the exact same thing. I won't talk about mini satellites or VNTRs much. These were bigger variations, stretches of hundreds of base pairs. We can't really look at them well with PCR, but they used to be looked at by Southern Blot, and they're just not used that much anymore. Okay, so now let's talk about SNP, single nucleotide variants. SNP is the common term, and I will slip and use that as a general term, even including things with allele frequencies less than 1%. This is a single nucleotide change. So one allele has a C, the other allele has a U. If this is more than 1% of alleles, it's technically a SNP. If it's less than 1%, it's technically a SNV, but again, they're generally lumped together and called SNPs. Some purists will try to use SNV. The vast majority are silent. The vast majority, 98.5%, fall in an intron, in between genes, don't do anything. Some will fall in promoters or other regulatory sequences, just like the thalassemias we talked about. These could happen to fall in a splice donor and cause a disease. They could occur in 5' and 3' untranslated regions. Again, around 1.5% are going to be in the coding sequence. Within an exon, part of it that codes for an amino acid. Well, what can happen then? We're going to talk about that in a couple of minutes. Well, when I first started out in the lab, when I was in training, there were only 3 this is chromosome 4. I'm showing this because this is actually the first gene to be mapped by genetic approaches was the Huntington's disease gene, which turns out to be out here. And these were all the markers that were available. These are the only variants in humans that we knew of that we could use to try to track the gene. Here's where they were by 91. Here's where they were by 96. If you now go into the SNP database, this is a year or so out of date, there are 4 million, over 4 million known common variants sitting on chromosome 4. Over 24 million known variants in the human genome. So there's lots of variation out there. In addition to these SNPs, to these single nucleotide variations, there are lots of variations in copy number. What this means, this is a big chunk of sequence that varies. And this was a real shocker. This was only discovered about 6 or 7 years ago. We had no idea this happened and was so common until we started looking at people's genomes. It was startling enough to accept that my genome and your genome had 3 million single nucleotide changes. You could take comfort in the fact that 98.5% of them are sitting in junk DNA. We don't have to worry about it. But here we find there are big chunks of DNA, 20,000, 30,000, 100,000 base pairs, that some people, it's duplicated. They have 2 copies right next to each other. Some have 3 copies. These are the location of all the common copy number variations, CNVs, in the human genome. Some of them as big as a megabase, 1 million base pairs. Many of them you can see by the length of these, but they're all over the genome. it's remarkable. So there's lots of large-scale variation, big chunks of DNA, often including a gene or a couple of genes that one of you might have. I've got 2 copies, 1 on each chromosome. You might have 3. 2 copies on one chromosome and the 1 on the other. That kind of variation is much, much more common than we ever had imagined. It does cause some disease, though, that we're beginning to discover, and I'll talk a little more about that later. Okay, so let's talk about the different classes of mutations. I've already mentioned this a bit in the globins. The different class of mutations that can occur if we mutate the coding sequence. So if we come along in here and have a SNP or a SNV, a single nucleotide change within an exon, what happens? Well, here you see an example. This is one class. We change the C to a T or U. GGC codes for glycine, and GGU codes for glycine. You know, there's a lot of redundancy in the genetic code. There are 64 possible codons, only 20 amino acids. Lots of examples of this. This is what we call a synonymous substitution, a silent change. It's a DNA variant, but has no effect that we can see. Keep in the back of your mind hemoglobin E. Maybe something like this might affect splicing or RNA stability or something else. But by and large, most of the time, we think these are generally silent. This is called a synonymous substitution or a synonymous SNP. No change in the coding sequence. Here's what's called a missense mutation or a non-synonymous SNP. This changes the amino acid sequence. Here we've changed that same codon. Instead of changing the C, we change the G to an A, and AGC happens to code for serine. And so we've now made a single amino acid substitution, a missense mutation, or a non-synonymous SNP. Now, this could cause disease. This could be a glutathione of sickle cell anemia. Or it could be a silent variation that really doesn't matter much, of which there are many examples. So it could be harmful, could be neutral. You can't necessarily tell by this. Here's another important type. It's also a class of non-synonymous SNP, but this is called a nonsense mutation. It's changed an amino acid codon to a stop codon. An A to a T, TAG is a stop codon. Protein translation stops here. You truncate the protein. This almost always, not always, this almost always causes disease. This is generally, particularly if it's early on in the protein, going to disrupt the protein and result in a loss of function. So you can't be sure, but you can usually assume that this is a disease, potentially disease-causing mutation. Here's a frame shift mutation. Here we've inserted or deleted a nucleotide or a couple of nucleotides, a small indel, insertion deletion. If it's not divisible by 3, it's going to change the reading frame. So now, you know, you had U, and you insert, delete one of these U's here. This is a deletion of a single U. Everything slides over, and you change the way you read this. You change the amino acid. Eventually, by chance, you generally encounter a stop codon. So generally what happens with a frame shift mutation is at the point of mutation, you have garbage for a while, and then it stops. And these also almost always cause disease, but not always, or potential to cause disease. Okay. Splice site mutations, we talked about plenty of examples already. These will alter the RNA sequence by affecting splicing, just like the examples we showed in hemoglobin. There are lots of other types of mutations. We can have mutations in regulatory regions of the gene. We can have large deletions or insertions, like the examples that we saw in alpha thalassemia. We can have translocations or inversions where we stick one part of one chromosome next to part of another chromosome. Lots of things can go on. And again, Murphy's Law, any one of these things you can imagine has happened. Sometimes associated with disease, sometimes not. Okay. What about these copy number variations, deletions of reasonably small regions of the genome? Up until modern times, up until the past, 5-10 years, the only way we could look at chromosomes was cytogenetics. We'd actually look at the chromosome under the microscope and stain it. And if a big piece was missing, you could tell. If there was an extra chromosome 21, oh, look at that. There are 3 chromosome 21s. There are only supposed to be 2. We were pretty good at telling that. That's Down syndrome. And usually big things like that, an extra chromosome, missing a chromosome, are all associated with disease. Missing a big chunk of a chromosome, usually associated with disease. Well, now that we have these incredibly powerful tools, we're finding that there are these little teeny changes that are sometimes associated with disease and sometimes aren't. This is very rapidly moved into clinic. This is now a standard procedure in the genetics clinic, not really anywhere outside of the genetics clinic. What you're looking at here is a hybridization chip that's looking at the whole genome. And if the signal ends up here, that means that part of the genome has the expected number of 2 copies, one from mom, one from dad. If it's above, that means there's an increased copy. If it's below, that means there's a deletion. And you can see in this position, we've only got 1 copy rather than 2 for this region of 5 or 10 or so million base pairs of the genome. So we can do this kind of survey in any individual now as a diagnostic test. And it turns out, if you take kids who come into the pediatric genetics clinic with some kind of profound intellectual disability that's unexplained by any other diagnosis, you will find a major copy number variation like this in 10 to 20% of them. Are they causative? We don't always know. There have been a few now that have been seen repeatedly in children with severe disabilities and never in normals that we're pretty sure are causing the disease. The problem is you will find some variation like this in all normal people. Not usually big ones over many megabases like this, like you find in 10 to 20% of them. But if we did all of us, we will all have some copy number variation, some well-known normal variation, some novel we've never seen before. Is this a silent variant, a new variant in you, or is it causing something? Not always easy to tell. Yes?
Why do the band lengths differ in each?
What do you mean Oh, this is just the readout of the test. This is just telling you how the signal looks compared to normal. So the amount of signal here compared to a normal control. There's less signal for this part of the chromosome 9. That means a part of it's deleted. There's variation within the test, and within the red bars is a normal variation. It's not worth knowing the details of how this is done, but this is what the readout actually looks like from these tests. And we'll see it when you get into the clinic, and it will be different and a different readout by the time you guys are doing this. Okay, so in the last couple of minutes talking about this, let's think about this issue, which I've referred to a number of times. When we find, and this is an increasingly challenging problem in the clinic. Patient comes in, you see them, you do a test, and you get a result back. There are basically 3 possibilities. It could be completely normal. It could be a clear cut, we know the DNA change. We thought they had sickle cell anemia. We do the test, we find the sickle cell mutation. They have sickle cell anemia. Easy to interpret. But there's a large gray area. We find a mutation. We don't know. Could this be causing the disease? Could this not be causing disease? For example, and this happens all the time, let's say a woman comes in to see me in the genetics clinic who has a very strong family history of breast cancer. Mom died of breast cancer, a sister who's died of breast cancer, an aunt who had ovarian cancer. Well, there's an autosomal dominant familial cancer syndrome associated with mutation in either the BRCA1 or the BRCA2 gene. Well, she'd like to know, because if she has the mutation, she got that from her mom, she has a 50-50 chance, it's autosomal dominant, she might like to have a prophylactic mastectomy or have her ovaries removed or various things that people do in that situation. Obviously, she doesn't want to do those if she doesn't have the gene. Can we tell? So we order a test, spend a lot of time talking to her about it, go through informed consent, we send off a DNA sample, and it comes back with a mutation that it identified in the BRCA1 gene. What do you tell the patient? Well, you got to be really careful. Let's say it's a nonsense mutation in codon 2. Well, you can tell her, that almost certainly messes up the BRCA gene. This is probably your family's mutation. You got it. Perhaps it's a mutation changes an alanine to a glutamic acid in codon 56. Well, this has been seen before in 20 women who had breast cancer who have BRCA1 in their family, and this was it, and never seen in 10,000 normals. Well, that's probably a real mutation. But here's the tough one. She's got a change in codon 43, alanine to glycine. It's never been seen before. This happens a lot. So what is it? Is that her family's mutation, or is this just a silent variant? What do you tell her? And there's no right answer. This will be called in the clinic and on the readout from the lab as a variant of unknown significance. And it's a really difficult thing. And of course, whenever you do a test now, you have to think about the possibility that that might be the outcome and how you're going to deal with it. So how do we detect mutations? I used to, again, spend whole lectures talking about this. Right now, almost all by direct sequencing. And the methodologies are changing all the time. The real question now is, we do direct sequencing almost always now. And you basically, there are three flavors now, and this is rapidly changing. Now in the clinic, usually, this woman has BRCA1 and wants to know whether she's got a BRCA1 mutation. We'll generally sequence just the BRCA gene. But very soon, we'll do the whole genome or some part of it, and we're starting to do this. You can do just the exome. There's methodologies to take just the exons and sequence all the exons, all 20,000 genes. Well, of course, that includes the BRCA1 gene. Right now, there's a company that has a patent on the BRCA1 gene called Myriad, and they charge a lot for it. And it ends up costing $1,500, $1,000 or so dollars. There are a bunch of rare genetic neurologic diseases, and a company called Athenia Diagnostics has the market for that and patent protection. And they'll charge a few thousand for each test. And you might test 5 or 10 different genes and rack up a bill of $10,000, $20,000 of genetic testing. You can do the whole exome now for under $1,000. Well, if you could all do all 20,000 genes for under $1,000, why would you want to do all these individual tests? And I'm sure by the time you guys are practicing, this is what we'll be doing, or maybe the whole genome when that gets cheap enough. Of course, the nice part about the genome is you only need to do it once it's been sequenced. And we made that BRCA1 diagnosis. When the woman comes back 5 years later and wants to know about her colon cancer risk, we don't have to order another test. We just look in the computer. Yeah?
Do you expect patch detections on genes to get held on to go away?
I sure hope so. Obviously, I'm expressing my bias here. I think they will. They already have in Europe for breast cancer, and it's an ongoing battle. I think it will. I'm sorry. The question was, what do I think is going to happen to the patent control of can you patent genes for tests? This is a hot ethical issue in genetics. I think the vast majority of us, I think almost every academic agrees it should go away, and I think we're all pretty convinced it will fairly soon. They're just not going to be able to control this. When you do whole exome sequencing, they can't control that, and when you have the data, it's probably just not going to be possible to control you using it. So I think it will go away. Yeah.
In the breast cancer case you talked about, is there any reason that you couldn't get the DNA from the family and sequence theirs?
Well, great question, and we'll talk more about this in other lectures. Absolutely. That's really what you want to do. I'm not getting into that here, but the yet bigger question that you're touching. The question is, what about doing the family? Wouldn't that help? The answer is absolutely. Because let's say you find nothing in her gene. Now what do you tell her? Well, could be that we just missed a change in your family. It's in the middle of an intron somewhere. We don't know what it is. Or your family has a mutation we could have found in your mom, and you didn't get it, and you're normal. But we don't know which of those it is. Well, that's helpful. If you test just the patient, the bottom line is all you can do is give her bad news. But if you test her mom first, you find her mom's mutation. Now you test her. She either got it or she didn't. Now you can tell her, yes, you have it, or no, you don't. If you can't find mom's mutation, you don't bother testing her. That is by far our preferred route in the clinic. You always want to start out testing a clearly affected individual if you can. Identify the familial mutation. And again, eventually this is going to become standard. But that's, getting ahead, but it's a key question. Okay. So how can you know for sure that you're dealing with the real thing? Obvious disruptions of the genes were generally safe, assuming a nonsense mutation, a big deletion, rearrangement, a frame shift. Usually you'll assume those are the real deal. Sometimes we can, if you know what the protein does, for example, patient has hemophilia, you test the factor VIII gene, you find an alanine to the glutamic acid. You now make recombinant protein in the laboratory that has a glutamic acid instead of an alanine and show it doesn't work anymore. Well, okay, that's the real deal. In practice, that's not that easy to do for very many things. This is an important one. If you have a standard genetic disease, like, say, neurofibromatosis, autosomal dominant, child has it, neither parent does. This could be a new mutation. You'll learn more about this later, but 70 new mutations in each one of us. If one of them lands in a gene like NF, you might have neurofibromatosis. How do you know that the child is a new mutation or not? Well, if no one else in the family has it, parents don't, their genes are normal, and you find a mutation in his that wasn't in the parents, you can be pretty sure that's the real deal. Because what's the chance that one of the 70 new mutations he got just happened to land in that gene, that one of 20,000 genes, and he's the first one in the family to have it. It's a subtle point, but more and more that's going to help us. There's been some use of this now in diseases like autism, where there have been a number of cases found where there's a significant new copy number variation in the child, not present in either parent. And the child has autism and neither parent does. And at least some of those we think are actually disease-causing. We use computers to predict, based on the structure of the protein, we think this might be disease-causing or not. This is very, very imprecise. And it's a little help, but not a lot. And at the end of the day, you're often left with this, not knowing for sure. Is this a rare disease-causing mutation, or is this a private polymorphism rare variant? And keep that in mind, because you don't want to make the mistake of calling one the other in either direction. And often for now, we're left saying, this is a variant of unknown significance. I don't know what to do. Might be your disease mutation, might not. Okay, so hopefully you're comfortable with the concept of polymorphisms. What that means, the different types, in particular the ones you really need to know about and understand are these simple tandem repeats that have many different alleles, so we can use them, forensics and things, SNPs or SNVs, single nucleotide changes, and copy number variations. And then the different classes of mutation, which we've covered here and in the globins. And then this fundamental idea of when you find a variant, we've got 3 million of them, each of us. A good chunk of those are unique to us. Very few other people have. 70 of them nobody else has. How do you know when that is disease-causing and whether it's just nothing in particular? And that's a fundamental issue we've got to keep thinking about. Okay, we'll break till 11:10.